Good evening. The last class we discussed the Bryophyta, and those are first land plants and where the vascular system is not developed, so those are non-vascular cryptogams. The next group, next higher group, that is Pteridophyta. In this Pteridophyta, there you can find a true stem, leaf and roots, and these plants having vascular structures like xylem and phloem, so these are vascular cryptogams. And another name that is given, the plants having the tracheids are called Tracheophyta. So among the trachophytes, these are the primitive type of tracheophytes. So the word Pteridophyta was coined by Haeckel. The meaning is pteron means feather, phyton means plants. That is, the plants having feather-like leaves are called pteridophytes. So generally, as I told you earlier, the bryophytes are called mosses and pteridophytes are called ferns. Actually, fern means higher pteridophytes. They are having very big leaves. Those are called the fronds. They are just like feather. So these are called plants having feather-like leaves. And the study of Pteridophyta is known as pterology or pteridology. Both are accepted. Just like algology and phycology, here you have two names, pterology or pteridology. In the same way, the scientists are called pterologists or pteridologists. Professor P.N. Mehra was the famous Indian pterologist, and remaining international pteridologists are Scott, Church, Ames, Campbell, Zimmerman and Bauer are the famous international pterologists. Regarding the characteristics, the sporophyte is the dominant stage in the life cycle, whereas in Bryophyta, gametophyte is the dominant phase. In this life cycle, there are two independent generations. In Bryophyta also, you have two independent generations, but whereas the sporophyte is independent upon the gametophyte, whereas here two generations are independent, morphologically dissimilar, and the sporophyte is divided into stem, leaves and root. Root system is adventitious type. The stem is similar, the structure that of the seed plants, the structure having the vascular bundles, the element phloem, etc., similar to higher plants. Many are provided with a horizontal or oblique rhizomatous stem, that is horizontal on the below the stem, and some are straight. Lycopods and horsetails, there are aerial stems which reach several feet height. They are straight, and some of the lower forms are horizontal and little higher forms are vertical and straight and long. So here you can find two diagrams are given, the lycopod and equistem. There the stem is very strong and very height. These are the first land plants having vascular tissue. I told you already, these plants are vascular cryptogams having vascular structures. Xylem mainly consists of tracheids and phloem consists of sieve cells without companion cells. In higher plants, angiosperms and dicots, etc., where you can find the xylem consists of tracheids, vessels, etc., vessels, and phloem consists of sieve tubes and companion cells. Whereas here it is just primitive than angiosperms. It consists of only tracheids and the xylem and sieve cells without companion cells you can find in pteridophytes. Next, the leaves are usually most conspicuous structures. If you happen to see the plant, the whole plant looks like a very big having leaves. And in lycopods and horsetails, they are typically microphyllous. They are very small. The leaves are very small, so those are called microphyllous, that you can see later. In ferns and pteridophytes, they are macrophyllous, often large. They are simple or compound, and the leaves of these ferns are called the fronds. Now you can see these two types of microspillous plants and macrophyllous. In the first diagram, then lycopodium, you can find the leaves are very small here, very small leaves are circularly and just spirally arranged on the stem. And whereas here you can find in Glycinia, this is a fern where you can find very big leaves are there. So these are called macrophylls or fronds. The young leaves, the very interesting thing that you can find, the young leaves of most of the pteridophytes, especially the ferns, exhibit a circinate vernation. Vernation is a technical term, that is the arrangement of the leaves in young condition is known as vernation. Perhaps you might have known venation. Venation means arrangement of the veins in the lamina, that's called venation. Here it is vernation, that is the arrangement of the leaves in its bud condition is called vernation. And you see how it is just looking like a clock spring like twisting of these leaves. This is known as the circinate vernation. It is one of the characteristic feature of the ferns. Next, large leaves. Large leaves of the ferns are called fronds. Large leaves are called the ferns. The reproduction normally takes place by means of spores produced in sporangia. The sporangia are born either on the leaves or on the axis. The sporangia may be on the leaves in the lower forms. The primitive forms you can find the stem itself where that one, and whereas in little higher forms you can find they are developed on the leaves. So the sporangia bearing leaf is called sporophyll. Phyll means leaf. Sporophyll, sporangia bearing leaves. Here it is two species are given, two genera. Here you can find this is Selaginella, where you can find the sporangia are formed in the axis of the leaves, and whereas here at the end of the branches you can find the sporangia, it is in Rhynia. Next, sporangia bearing leaves are called sporophylls. I told you already there are two types of sporangial development. How the sporangial development? One is leptosporangiate, another is the eusporangiate. In the leptosporangiate, the sporangium develops from a single superficial cell. Develops from a single cell. Example, Glycinia, Marsilia, Neprolepis. The second one is the eusporangiate type. The eusporangiate developing from a group of cells. The sporangium developing from a group of cells. Example, Lycopodium, Selaginella, Equisetum. Next, when the spores are of the same size, the plant is called homosporous. Lycopodium, Neprolepis are the examples where all the spores are of same size, same color, same structure. So those are called homosporous. If it is having two different types of spores, bigger spores, macrospores, smaller spores, microspores, that is called heterosporous condition. When two different kinds of spores are developed, this plant is known as heterosporous. Example, Selaginella, Salvinia, Marsilia. Now let us see these two types. So here you can see in the first diagram Selaginella, where you can find some are small spores and some are bigger spores. So the smaller spores are called microspores and bigger spores are called the megaspores. Whereas in the Lycopodium you can find all the spores are of same size and color, so it is homosporous condition. Next, the small spores, as I told, the small spores are called microspores, which are produced in microsporangia, while the large spores are called megaspores, which are produced in the megasporangia. And these spore mother cells undergo, because the Pteridophyta, it is a sporophyte. Remember, sporophyte is always diploid, gametophyte is always haploid. So the diploid sporophyte here gives the spore mother cells, they undergo reduction division and form haploid spores. So these haploid spores on germination develop into gametophyte. Gametophyte is also known as, this is another new things that you have to remember, the gametophyte is called the prothallus. That gametophyte of pteridophytes, prothallus, and it bears the sex organs. It is haploid. These sex organs, look here, this is the prothallus of pteridophyte, prothallus, and in this prothallus, just it is a heart-shaped structure, and this here apical side, there is a notch is there, that is called apical notch. In the lower side, you can have a number of rhizoids for absorption purpose, and the archegonia are female sex organs develop around the apical notch, and antheridia they are developing near these rhizoids, near the rhizoids. So the sex organs have a sterile jacket layer. The male sex organs are called antheridia and the female sex organs are called archegonia. The antheridium produces many spirally coiled multiflagellate antherozoids. So the antheridium, there is a male sex organ, it gives more male gametes. Those male gametes are multiflagellated. Their gametes multiflagellated. And the archegonium having, I told you already, the archegonium having a flask-shaped structure, and this narrow portion is called the neck, and lower broad portion is called the venter. And in the neck you can have a number of neck canal cells, and in the venter you can have a small venter canal cell, and you have a very big egg cell is found. So the fertilized, so the male gametes enter into the archegonium and fuses with the egg. In the meantime, the venter canal cell and also the neck canal cells are disintegrated and leave a cavity here, a canal here. So a number of male gametes enter into it, and finally one male gamete fuses with the egg and form a diploid zygote. The fertilized egg becomes the zygote. So this zygote develops into an embryo which gives rise to diploid young sporophyte. So you start with a diploid young sporophyte, again it gives the haploid what's called gametophyte. Gametophyte are prothallus. There you can find male and female structures, male and female gametes, they fuse and again give the diploid sporophyte. So it gives to diploid young sporophyte. The sporophyte is large, long-lived and independent when compared to bryophytes. Whereas in bryophytes, the sporophyte is a parasite, or sometimes it is attached with the gametophyte only. Whereas here they are independent. Gametophyte and this sporophyte are independent in pteridophytes. Since the diploid sporophyte and haploid gametophyte are morphologically dissimilar, the pteridophytes show heteromorphic alternation of generation. Here the gametophytes are very small, as I told, this prothallus, a horse-shaped one, and very small, whereas the sporophytes are very big. So here you can find the different size and structure, so heteromorphic alternation of generations that you can find in pteridophytes. So regarding the classification of pteridophytes, G.M. Smith 1955 has classified pteridophytes into 4 divisions. Number 1, Xylophyta, example Xylotum rhynia. Lepidophyta, example Selaginella lycopodium. Calamophyta, example Equisetum. Pterophyta, example Neprolepis adiantum. Now let us see some few characters of different divisions. We see the first one, Xylophyta. This is Xylophyta. The sporophyte is rootless, but rhizoids are present. Sporophyte is rootless and the stem having the rhizoids so that it can absorb. Stem contains vascular tissue, that is a protostele. I will tell you later. It is a simple type of stele where you can find a group of cells of xylem surrounded by phloem. This is called a protostele. I will tell you later. And central solid of xylem surrounded by phloem. This is called the protoxylem, protostele. And regarding the Xylophyta, the leaves are absent or leaf-like appendages occur spirally on the stem. Spores are produced in terminal sporangia, which borne singly at the tip of the branches. At the tip of the branches, the sporangia are developed. And the spores are homosporous. Here you can find these are homosporous. Spores are of same type. Here you can have 2 examples. One is a Xylotum, another is a rhynia. Rhynia is a fossil, now not available, and the Xylotum is available. Those 2 photographs you can see now. So here you can find this is the Xylotum living and having a fork-like branches. At the end of the branches you can find the sporangia. And here you can find the rhynia. This is a fossil. It is also having the sporangia at the tips. Having protoxylem, you can have here, you can find protostele. So the next group, Lepidophyta, the plant body is differentiated into root, stem and leaves. Leaves are small, microphyllous, simple. The vascular cylinder is a protostele, primitive type. So slowly in the divisions also you can find a variation and evolution you can find from slow development to higher development of xylem, etc. The vascular cylinder, it is also protostele here you can find, and sporangia borne singly on the upper surface of the sporophyll or its axis. So the sporangia are developed in the axis or on the upper surface of the leaf. Here you can find the leaves, upper surface, addoxial surface, or at the margins also you can find. Generally the sporophylls are aggregated, form cones or strobili. So sporophylls, a number of sporophylls are compactly arranged at the tips of the branches and form a cone-like structures. So those are called cones or strobili. Example, Lycopodium, Selaginella. So here you can develop this one. The end of the branches, the Lycopodium is given, homosporous structure, branches given. There at the end of this branch, you can find the cones are there. If you happen to cut in this cone and see through microscope, here you can find having a similar type of spores, a homosporous condition. Next, this is the Selaginella. There you can find these are the branches. At the tip you can have the cones are here, and if you take a transverse section, there you can find some spores are very big, it's called megaspores, and smaller microspore, so it is heterosporous condition. The third the division, that is Calamophyta. The sporophyte have true root, stem and leaves. The stem is jointed with distinct nodes and internodes, just like looking like a sugarcane plant, and the internodes are hollow, longitudinally ribbed and furrowed. It is the equisetum. The thing is, here you have to remember the stem is jointed with a distinct nodes and internodes. That is very important thing that you can see in the next photograph. So here you see as if it is a small type of a sugarcane plant. There is here you can have this node, another node, internodes. Nodes and internodes are there. This is a tall plant like this. You can find a stem with nodes and internodes, you have to observe. The both protostele and siphonostele are present here. Another new term here, I told you already in the first time, it is a protostele. Central xylem surrounded by phloem is a protostele. Whereas the siphonostele, when the pith is found in the center, and around this you can find xylem, another phloem, this is called a siphonostele. Siphon means just like a tube, where the pith is there in the center, it looks like a tube, it is a siphonostele. Detail I will tell you later. And the leaves are scales like, and sporangial development and the sporangiophores form cones or strobili. These also you can find strobili or cones. The spores are homosporous, the same type of spores you can have, whereas in this there are two examples are given. One is equisetum, that is living, and sphenophyllum, this is extinct, not available. Some years back it is there, but it is not there, so it is called extinct. So here two diagrams are given. One is the equisetum, this is living, you can find here nodes and internodes in a small plant, nodes and internodes, and a bunch of leaves you can find. Whereas here this is extinct, sphenophyllum. Here also you can find the leaves are arranged around the stem as a circle and having the cone at the tips of branches. The fourth one, Pteridophyta, where the sporophytes have true stem, root and leaves. These are highly developed pteridophytes. The Pteridophyta are generally called the ferns, highly developed pteridophytes. The leaves are large, described as megaphylls or fronds. You can see the leaf. Fronds, these are called the fronds. You see how they are very big leaves, very big leaves. So these are called the fronds that you can find in Neprolepis species. Leaves are very big, megaspore, megaphylls. And the stem is protostele, siphonostele, dictyostele, and sometimes polycyclic stele. These all I will tell you later. So these are all the types of steles you can find in Pteridophyta. Sporangia are developed either upon the margin or upon the abaxial side, or abaxial side, upper side of the leaf, or it may be in the edges of or margins of the leaf. The sporangia are developed. Bunch of sporangia are developed. So here you can see Here the sporangia are developed in the abaxial surface, and where you can find long stalked sporangia are developed. Here you can find it is on the margin the sporangia are developed. Both types are present in these ferns. Mostly they are homosporous. Example, adiantum, and a very few species are heterosporous that you can find in Marsilia. So both types are present here. So adiantum is given. Here you can find the long leaves here, and this Marsilia you might have seen near water. Near water you can find, and there everywhere you can find 4 leaves are there. 4 leaves at the tip of this petiole you can find 4 leaves. This is Marsilia. So as I told you, this is the stellar evolution. How the steels are developed. How the steels, with the steel is nothing but. So it was proposed by Van Tygem and Dowlett in 1886. It is a new thing that you have to learn. But so far you can remember the bryophyte, algae, fungi, etc. But the stellar evolution or stellar system is a new topic that you have to learn carefully. So it was proposed by Van Tygem and Dowlett in 1886. According to them, the steel comprises of xylem, phloem, pericycle and the pith wherever it is present. So a steel consists of xylem and phloem surrounded by the pericycle and endodermis, etc. So you remember the internal structure of dicot stem, dicot root, monocot stem, monocot root, etc. so that you can understand. So now let us see the types of steels. The first type is the protosteel. I told you already in the lycopods you can find the protosteel. It is very simple and primitive type, and other types have been developed from this only. All the others developed from this one, but this protosteel is of 4 types. So the first one, that is the haplosteel. Haplosteel, a solid center smooth core of xylem surrounded by phloem and then pericycle. The center xylem, above the xylem phloem and pericycle and endodermis. So this structure, it is the haplosteel, is the primitive type and remaining developed from this only. This type you can find in Rhynia and Selaginella crassiana. Selaginella crassiana and Rhynia you can find this type of steels. The second one is called this actinosteel. Actinosteel, here you can see clearly the protosteel and central xylem is radiating here, radiating like this, and in between you can find the phloem. So a protosteel with the central xylem of radiating arms surrounded by phloem and pericycle. This is called actinosteel. Example, Xylotum and Lycopodium serratum. Serratum species, you can find this type of actinosteel. Next, plectosteel. Plectos means plates. So here the xylem is present in the form of separate plates. Separate plates, xylem, separate plates you can find, and phloem is present between these xylem plates. Between these xylem plates, the phloem is present. So here it is xylem, and in between the phloem you can find here phloem. That is called a plectosteel, where xylem is in the form of plates and phloem is present in between. Next we have this mixed protosteel. Mixed protosteel, what happened here? The xylem is present in the form of discrete units. Xylem is divided into different groups, and in between phloem is present. The xylem is present in the form of discrete units embedded in the phloem. Example, Lycopodium cernuum. Lycopodium cernuum, you can find the mixed protosteel. The second one is a siphonosteel. I told you already, a protosteel having a pith is known as siphonosteel. Here the steel, a central pith surrounded by vascular tissue, is called siphonosteel. It's very important. A central pith surrounded by vascular tissue, that is xylem and phloem, is called a siphonostele. The origin of pith may be intracellular or extracellular. This is of different thoughts. The formation of xylem in the center, that is, the central cells may be modified into xylem or form into the pith, or due to the leaf traces, the cortex enter into the pith. Cortex enter into the center and form as pith. Different theories are there. You need not worry. So next, the siphonosteles are of two types. One is called ectotrophic siphonostele, ectofloic siphonostele. Ectofloic means phloem is found outside the xylem. So here you can see this is the xylem, center xylem, and this is the phloem, outside phloem. So this is ectofloic siphonostele. Xylem is surrounded by phloem. This type, that phloem is present on outside the xylem. Example, Osmunda and Shaigia. There you can find this type of ectofloic siphonostele. The next one is siphonostele. This is amphiploic siphonostele. Amphi means both sides. Here the phloem is present on both sides of the xylem. You look here, the center, there is a pith is there in the center, central pith. This is surrounded by phloem, and again xylem, again phloem. So phloem is found on both sides of the xylem. That is called amphiploic siphonostele. Amphiploic siphonostele. You can see the labeling also there. You can see central pith you follow, and then you can find phloem, and then xylem, and then phloem. So it is given as inner phloem, outer phloem. So example, Adiantum and Marsilia, you can find this amphiploic siphonostele. The next one is the third one, that is called solenostele. Solenostele, a siphonostele with leaf gap. A siphonostele, yes, you can have, the siphonostele is there, but it is having any leaf gap. It is a cutting takes place in the xylem, and you can find this solenostele. This is of two types. Solenostele, ectofloic solenostele. So look here, because of the leaf gap here, it is cut, and it is also two types, ectofloic. Here also xylem, above the xylem, outer phloem is there. This is ectofloic solenostele, where the phloem is present on outside the xylem. Example, many ferns you can find this type of ectofloic siphonostele. The next one, amphiploic siphonostele. Here also, because of the leaf gap, here you can find, and inner phloem, middle xylem, outer phloem. So inner phloem, outer phloem. Now in between you can find xylem. This type is called amphiploic solenostele, where the phloem is present on both sides of the xylem. Example, Marsilia, Adiantum, you can find this type of phloem, this type of steles. Next is called the fourth one, dictyostele, where you can find in the siphonostele, because of a leaf gap, you can have this vascular structure, vascular cylinder is cut. But whereas here, overlapping leaf gaps, because of many leaf gaps, the stele is cut into different smaller units. So each unit is called a meristele. Each unit is called meristele. In this, the vascular cylinder breaks up into number of smaller units called meristeles. So in a dictyostele, dictyos means cut into pieces, and each piece or each unit is called meristele, that you can see now. So look here, here these are a separate units. Here you can find this is one unit, one unit, one unit. In each unit, here you can find a central xylem surrounded by phloem. These are called meristeles. Each meristele is protostele, just like a protostele in nature. Pteris and Adiantum, you can find this type of stele. Now another type is called polystele. Polystelic condition. When more than one stele is formed, it is known as a polystelic condition. For example, in Selaginella, you can find this is one stele. There you can find central xylem surrounded by phloem, pericycle, endodermis. another stele. In the same way, you can find 2 or 3, sometimes you can have, those are called polystelic condition, example Selaginella. The last one, that is called polycyclic steles. Polycyclic steles means the steles may be arranged in 1 circle, 2 circles, 3 circles, etc. When the meristeles are arranged in more than one ring, it is known as polycyclic stele. For example, in Pteridium there are 2 rings, and Matonia there are 3 rings are found in the xylem. So these are called the polycyclic steles. Polycyclic steles. Okay. So far you have studied the general characters of these pteridophytes, their classification, and stele or evolution. So remember, in pteridophytes, the dominant plant is sporophyte. It gives gametophyte, and alternation of generations or heteromorphic alternation of generation. Whereas in Bryophyta, the dominant plant is gametophyte. It gives a sporophyte. And in this sporophyte here, in Bryophyta, that is dependent. Whereas in pteridophytes, both are independent life cycles you can find. Thank you.